On behalf of the Crossroads at Big Creek Learning Center and Nature Center, I welcome you, everyone, both here and in person and those joining via Zoom, to our third Fishtails lecture this year, where we present the science of Great Lakes fisheries. Tonight, we will learn about using environmental DNA, also called eDNA, as a tool to detect the arrival of invasive carp in Lake Michigan Basin by Dr. Cari-Ann from the U.S. Fish and Wildlife Service. The Fishtails Lecture Series is an educational program here at Crossroads, and my name is Mark Holy, the Fishtails program organizer. I want to thank the Door County Library once again for their partnership to present our lectures via Zoom and Facebook Live for those who cannot attend in person. I want to mention again, we have one more Fishtails lecture on our final one on March 17th, when Dr. Karen Murchie, the Director of Freshwater Research at the Shedd Aquarium, will present her research on white and long-nosed suckers in the Great Lakes. Dr. Murchie's sucker research does span the entire Great Lakes, but the Door County tributaries are a focus area for her, and so you will not want to miss it. There are a few housekeeping details for those joining via Zoom. Your microphones will be muted or should be muted during the presentation. Please use the chat feature to ask questions, and we'll address those at the end of the presentation. This lecture includes live inscription. To access that feature, please click the closed caption symbol in the bottom toolbar, then click show subtitle. And then finally, this lecture will be recorded and available at the Door County Library calendar website. Now, in case there's people you know that tried to access the last lecture on acoustic technology in the Great Lakes by Dr. Kruger, the library has been pretty busy in the month of February with the big read and so on, so that's not quite ready yet, but be sure to check back every once in a while because it soon will appear. And we can't thank them enough again for doing that. Tonight, we're actually trying out the OWL technology and everything, so hopefully it all works. I know we had a little bit of glitch last time, but it's going very well tonight, which should improve the presentation. You know, when you catch a single rare or invasive species for the first time in a water body, it usually is not the only fish present because our standard sampling techniques can be very site-specific and somewhat limited in scope. You know, in the case of invasive carps that are slowly making their way towards Lake Michigan in the Illinois River, the use of forensic technology to sample carp eDNA or carp DNA in water samples has been adopted by Great Lakes managers to enable us to detect their presence even before we can catch them with standard sampling gear. We are fortunate to have Dr. Cari-Ann here. She is the manager of the Environmental DNA Surveillance Program for Lake Michigan for tonight's presentation. Cari-Ann grew up in Miami, Florida, but she spent summers in Hayward, so she has always claimed Wisconsin as her home and has always been a diehard Packers fan. And I think she has a picture of it later on, but during those summers at Hayward, she actually developed into a world champion log roller, and one of the youngest ever to do so. She attended Auburn University and completed a degree in wildlife science, and eventually got a position with the state of Alabama that let her return to Auburn to receive a master's degree in fisheries. After graduating, she accepted a position at the Cooperative Fish and Wildlife Unit at South Dakota State University, studying rare and endangered streamfish throughout the Dakotas. Out of concern for the encroaching invasive carps, Cari-Ann developed a PhD project there to study trophic and invasion dynamics of silver and bighead carp in South Dakota. With her PhD in hand, she went to work for the USGS Columbia Environmental Research Center in Missouri, where she continued her research with silver, bighead, and grass carps throughout the Mississippi River Basin. And then in 2016, she jumped at the chance to return to Wisconsin and join the U.S. Fish and Wildlife Service at the Green Bay Fish and Wildlife Conservation Office to lead the service's aquatic invasive species early detection and monitoring program across the Lake Michigan Basin. And she was the last major hire I made before I retired. So let's give a warm welcome to Cari-Ann Hire.
Okay. Hello, thank you.
Should do.
All right. Well, it's good to be here. I'm excited to be speaking about some of the work that I do. These kind of presentations or talking to you guys is always a very wonderful change to some of the more sciencey, technical presentations that we always have to give. So this is really good to be sharing what I do with you all. So with that, Mark kind of stole my thunder here on the beginning of my presentation, but so like he said, I'm dual citizenship, Florida and Wisconsin. I will say I don't really claim Florida. I don't like Florida. I like the winters. I like trees. I like water. I like fishing. I like wildlife, everything like that. So this thing is falling off. And so when I first started out, I knew I liked the outdoors. And everyone in fisheries or wildlife always says, I like the outdoors. You know, why did you become a fish biologist or a biologist? And I was maybe 8 or 9 years old in Hayward at my family's cabin. And I liked to catch tadpoles. And so I would go out and catch tadpoles and catch one and, hold it and have it as my pet for that day. And there were some ducks there feeding on some corn. And I thought, oh, I'm going to throw the tadpole into the middle of the ducks and the ducks are going to scatter and it'll be really funny. Well, the ducks ate my tadpole and I went chasing after it all over the yard and I cried for the whole day. And I always tell people that was my first ecology lesson, that things eat things. So that sparked my interest in the environment and what's going on. And so I started out in wildlife science. I worked with wolves and bears and elk, like all the really cool, charismatic fauna. And I did that. And I said, what now? What can I do now? So I got a fish job and I loved it. And I never looked back. So that's kind of my background. So as Mark said, I run the environmental DNA program here in Lake Michigan. It's a basin-wide program of all the other fish and wildlife conservation offices throughout the Great Lakes. And so today I'm just going to kind of go back to the beginning and tell the story of how eDNA kind of started, how this new technology, new methods started. What is eDNA? What does it mean when we get a hit, when you hear we got a detection? And kind of talk about the invasive carps, which are silver and big head carp. Collectively referred to as the invasive carps. There's other carps, but I'm not going to be talking about those today. So, all right. So we have a lot of invasive species in the Great Lakes, and it keeps going up. Currently, right now, there's about 184 non-native species that are established in the Great Lakes. These are aquatic species. We all know they ruin the habitat or degrade the habitat. They'll outcompete native species. They can damage, important fisheries, tourism, etc. $200 million annually are used to combat invasive species in the Great Lakes. So this is from 2009. So quite some time ago when carp started to move up the Illinois River and get close to Chicago and close to Lake Michigan. And so this is when it started that people started to be concerned about carp getting into the Great Lakes. And yes, I have seen this picture before. They jump like that and it's absolutely crazy. So when I talk about these carps, I'm talking about silver carp, and those are the ones that everyone probably knows about, the ones that jump up in the air that you see hitting people and jumping 5 feet out of the air. Grass carp are in the middle. People don't typically think of grass carp as an invasive species. It is. It eats grass. It eats vegetation. It in a pond, it can decimate the vegetation in a pond, like in a very short amount of time, but I'm not talking about those. And then we have big head carp at the bottom here, and those are also, both of the silver carp and big head carp are planktivores. They filter what's in the water, and they can eat 4 to 5 times their body weight in a day. So they're voracious feeders. Common carp, as we all know, are also invasive, but they've been around forever, and we don't talk about those that much. So where did the silver carp and big head carp come from? Well, they came over here in aquaculture facilities, wastewater treatments back in the early 70s. And they were quickly known to improve water quality in these ponds, and they were able to control algae in some of these wastewater treatment centers. They were also could be used as a potential food fish for cows, and in words are not coming to me for that, but they were like the greatest thing. But then flooding happened, and they escaped into the Mississippi River, and they very, very quickly were able to establish and spread and continue to spread and reproduce and spread. And now we are where we are. So this is kind of this will take you through from 75 to now. And the red dots are just where they were detected. And so it started out in Arkansas in 1975, 1985, we're getting we're moving up the Mississippi, the Missouri River a little bit more. 10 years later after that, we're starting to see quite a few down in Arkansas, kind of moving up to the Illinois, getting a little closer. 2005, starting to explode. 2010, getting really, really close to that Chicago area. 2015, not very long ago, again, they're just taking over. And then this is what we have as of last year. And so the Illinois River, I don't have a pointer, but right by Chicago, they're knocking on the door, basically. They are reproducing. The reproducing population is 37 miles from Lake Michigan. So that might sound like a long ways, but carp can move 200 miles in a day if they want to. So it's really not that far. So in Illinois, why are they a big deal? In Illinois River, it is ground zero basically for silver carp in the world, and they make up about 90% of the biomass of the fishes in the water. This is about 4,000 fish per mile, and so that's a lot of fish. $110 million towards efforts to control them, manage them, remove them. Many native fish have declined in abundance in their condition as well. Walleye fishery was really, really big in Illinois, and it's kind of went kaput because of the carp. Also, it's a big commercial fishery in Illinois, and this was also negatively affected. And on a side note, commercial fishing now is big in removal of silver carp and big head carp, and it's allowing the slowing of the movement of carp up into the Illinois River. So commercial fishing is really helping us. And this is just kind of a fun little presentation. This was up in South Dakota, some work that I did for my PhD. And it's just kind of fun. So the first part here, this is big head carp, and this is just showing how they feed. And you'll see their mouths are really, really big, and they just swim along and filter feed and feed and feed and feed and get plankton. And so they will outcompete other planktivores like gizzard shad or buffalo. They'll just eat all the food, and they congregate like this, particularly below a dam or some sort of barrier. And so, yeah, this is a really good, they're just feeding. And then when we sample for them where they are, it's pretty dangerous work. So this is just, we were out sampling, trying to get, you know, a population estimate, trying to get some fish, and this is what happens. I will say that one time I got hit in the back of my head, and I blacked out for probably a second. It was not a big deal, but I still blacked out. And I was so mad because the fish got away. So, but I mean, it's crazy. It's a really cool thing to see, but not cool because they're causing problems. So, yeah, it's fun, I'll say, to be outside with these things. And we would have competitions about how many can we get in the net when we're driving by? How many can we catch? There have been people with broken noses, broken arms. A lot of people have probably seen the video of people water skiing through carp infested waters, and yeah, they're pretty crazy.
Sport.
Mm-hmm. And this in South Dakota, this isn't a very abundant population. So there's a lot here, but this is nothing compared to Illinois. We've got an actor here. There you got one. Slow motion. So, and we're not really sure why they jump. This is just a side note. They say that it's the engine or the motor, the noise from the boats. And typically we'll have this two boat. They'll jump after the first boat goes by, and the second boat can do that. So, so it's fun. And they're big. So, all right. So we have the Chicago is where we're concerned with them coming in. The Chicago Sanitary and Shipping Canal was put in the early 1900s, and it connected the Mississippi River Basin with the Great Lakes Basin. And so that's no good, because, you know, different species are going to be in different ones, and it's just not good to connect them. So in 2002, Army Corps of Engineers decided to put an electric dispersal barrier there in the canal that was supposed to stop the spread of species going between the two basins. And originally it was for round goby. I don't think they did their research, because gobies had already moved past, and it was too late basically to stop them. So it became invasive carps, and that's the main goal of this barrier right now, is to keep the carps on the other side in the Illinois River. So this is just kind of showing the barrier there. So you have a couple of the electricity, and the boats can still, the ships can still go through, and fish will still go through. They, you know, so it's not like a complete, they're not going to get through at all. There is still dispersal of fish through the barrier. So it's not foolproof. Wish I had that's fine. So the, can you see that? The mouse? Okay, so the star here, that's where the barrier is. And that is 37 miles from Lake Michigan. And then this yellow stripe here, this is where the adults are, where the front of the adults are, and where they're spawning. So they're still, the population front is still quite a ways away from Chicago. However, the these dots up here, this is where we've collected some live carp. A live silver carp and a live big head carp have been caught in Lake Calumet, which is only a few miles from Lake Michigan. We have not collected any in Lake Michigan outside of the cause yet, or even any of the other lakes either. So they're coming, they're moving. There are some that have been getting through, but not many. So when we're thinking about invasive species, they're going to be rare in a new environment if we can detect them early, and that's the main goal, is to find them early before they have before they can increase in abundance, before they can spread, so we can do something about it. So we can potentially either stop them or control them, remove them. And so this is the main goal of my program, of most invasive species programs, is to detect them early so we can still do something. Because once they're established, once they're around, it's really difficult to, one, get rid of them and manage them. So this came out, it just says what I just said. So through prevention, though prevention should continue in management efforts, we should have coordinated early detection programs and eradication programs in order to control and combat these new invasive species. So that being said, we get to environmental DNA, which is why we're here. This was a new method that came out, and it was done by University of Notre Dame in 2009. And it wasn't a new method; it's been used previously, but people started to hone in on it and recognize the use of early detection and control of invasive species. So Notre Dame started developing the methods for silver and big head carp, and worked with the Army Corps of Engineers to start an eDNA program in the cause. And then in 2013, the Fish and Wildlife Service took that over, and all of the different Fish and Wildlife Conservation offices in the Great Lakes had a program for each of their respective lakes. So we do eDNA across all the basins looking for silver and big head carp. So what is it? It is genetic material that is shed from a fish or an organism into the environment. So it could be from scales or feces, mucus, urine, gametes, anything that could come off of a body. It could be, now we're talking about it in the water. That's the most common way of looking at eDNAs in the water. But it could be in the air, it could be in the sediment. And it will predict the presence of a target organism. Now I'll talk a little bit more about what that exactly means, but and so I'm not a geneticist. This is as specific genetics that I'm going to get, but it most commonly is there's different DNAs. There's nuclear DNA, mitochondrial DNA, which is what we look at for looking at environmental DNA. It's most frequently used. It's more stable in the environment, in that it has many copies in each cell that will repeat and allow us to map the species that we're looking for and get that detection analysis. So again, it sloughs off the fish and they break down, and the cells break down, and it's released into the water, hair, skin, feces, reproductive movements, anything that can come off of the fish. But there's a lot of different factors that can affect the amount of DNA that is in the water, the amount of DNA that's in the water, and also how fast that it might degrade, a shedding rate of how often it comes off the body too. So there's a lot of factors that go into us finally getting that number at the end that says this species is here. So water chemistry, salinity, pH, temperature, all have an effect on the eDNA, the shedding, the degradation, decay of the DNA in the water. And then you have other factors, the water velocity in the water. So if a fish is shedding and the flow of the river is at high stage, the eDNA is probably going to flush out, and you might not get as many copies of it in the water. So there's a lot that goes on with how we, quantify this DNA. So why is this? Why are we looking at this? Because there's a lot of unknowns about it, and a lot of people think it's a little hokey, and they have, because it's just not understood. So traditional sampling gears, for example, with silver carp, they jump. They're very evasive. We have a really hard time of getting them in the boat with a net. Now they'll jump in the boat, yes, but to be able to capture them with electrofishing or nets, they're very hard to get. And so the eDNA allows us to detect these fish that we might not be able to hold in our hands. So these invasive species, cryptic species, we're able to get with DNA. It's very good for low density and those fish that you might not catch because there's not very many of them there. It's very sensitive to finding the rare fish. You can also sample in habitats where you might not be able to get with a boat, or it might be highly vegetated and you can't set a net there, but you can take a water sample. You can take a water sample pretty much anywhere. You don't even need a boat. So it's very effective at detecting a species. So how can we use this, not just for invasive species, but we can, detect native fish. We can detect any species that we want, given some certain parameters, but it's good for native species studies to assess biodiversity. How many species are there without actually having to go out and physically hold them. It's non-invasive, so we're just taking water samples, and if we're dealing with an endangered or threatened species that we don't want to handle, this is a great way to do that. It's been used on amphibians and fish and marine animals. It can classify the health of a system. Earthworm diversity, for example, is a really good way to show ecosystem health, if a system is healthy. And so you can go out there and find how many earthworms there are, and then that'll tell you that it's healthy or not. So it can be used for a wide array of things. And so for invasive species, again, it's early detection. I'll say that over and over again. That's the most important thing. You can detect based on various vectors of introduction. So in places where there's a bait trade that potentially could have silver carp in their bait, you can go and test that water, and it might come out to have a silver carp in there, and then that's no good. So the bait place will probably get in trouble. You can detect in ballast water, because that's a big problem. That's a big vector of invasive species, is ballast water from all the ships. And so you can just go take a DNA sample and kind of know what might be in there. So again, it's been used with invasive bullfrogs, mud snails, crayfish, pythons, all sorts of, a wide array of animals. It can be found in the sediments too, of terrestrial and aquatic systems. Now, I won't talk much about the sediments, but the DNA tends to settle at some point, and it'll settle into the sediment, and it could be there for years and years, hundreds, thousands of years. And so you can get ancient information of animals that were around a very long time ago. And ocean, it's been very successful in the ocean. We can't go physically down to very, very deep and, places we can't go, but we can take a water sample and we can see what might be down there. These deep diving whales, we don't know where they go necessarily, but we can take a water sample and see if their DNA is there. It's been used for sea lamprey monitoring. There's 7 species of sea lamprey that are very hard to differentiate, physically identify, and you could use the DNA to identify which species it is. So if you're not sure, genetics will allow you to differentiate that. Also, the sea lamprey have a large distribution, and physically sampling takes a lot more time than taking a couple water samples here and there. So why is it good? I've kind of mentioned it, but you don't have to hold the animal you don't have to hold the animal. It's not, you're not directly observing. And that's one of the things why people are a little standoff about it, because you don't have it in your hand. And I'll talk about that a little later. But you can sample a very large area. You can take water samples a lot faster than you can take an electrofishing run or set a net. And so the top right is our electrofishing sites. So there's 30 dots on there, and each one of those dots represents a 15-minute electrofishing run transect. And then in the bottom left, you can see the distribution of our water samples is a lot more. There's 200 sampling points there. And so we did the lower left in one day, and it took us a week to do the upper right. And so we're able to get a wider spatial coverage to see if we can detect the species. So it's more cost efficient, and it takes less people too. So in times of rough hiring and low people resources, this is a good way to do it. And again, it's, you can detect those low abundant, cryptic, rare, difficult to capture species. And I mentioned it earlier too. So it's non-invasive, again, with endangered or species of concern, threatened species that we don't want to, move them around or hurt them or whatever. We can use the water samples. So I mentioned this also is in identification. So you can see the fish, these fish, they all look very similar, and they're different species. So if we're out in the field, and this happens a lot, unfortunately, but we're counting fish, we're measuring fish, we're going through them. Those 4 look very similar, and you have to be very, very cognizant of what you're looking for and what you're looking at. But with genetics, it's very clear. It tells you what they are. Also, it has been shown in many, many studies that you will get more presence, more species detected with eDNA than with traditional sampling. And that's been shown in a lot of different studies. Okay, this is sciencey a little bit. So shedding. So when they shed off of the animal, it could be from gamete release or breeding activities. And also they're going to be a little higher in abundance at that time. eDNA will increase in those times. So you potentially would be able to predict a spawning event if there's a rise in the eDNA at a certain time, particularly for silver carp, because they will spawn or move to spawn at a rising hydrograph. Like when it goes up, they move, and they want to go. And so, so that's one thing that we can do with silver and bighead is to, hone in on these spawning events. Another thing to note is that this is a site that we sampled, and we got eDNA and we did not get any fish. So that tells me that they were there. We just didn't detect it with our traditional sampling. And then here at the rise in the hydrograph, we detected eDNA and fish. So it's able to do a lot of things. This is just like a depiction of where the eDNA comes from and then where it goes. So like I mentioned, we've got, biotic things and abiotic things that go into, that are the sources of why it comes out and why it's different. So temperature will affect the amount of DNA that comes out, or it'll affect down at the bottom, the sinks, it'll affect how it degrades in the system. And then you have transportation, especially in a river system. It'll dilute in high water events. It'll move faster. There might be less in the water. But then if you don't have any velocity, it might sink and go down into the sediment. So all these things you have to take into account when you're looking at this eDNA number that you get at the end. So I've talked a little bit about it. There's a lot of disadvantages, a lot of unknowns. We don't know a lot. We're learning more and more and more every single day about how this can be used and how we can better predict and take our predictions and apply them to invasive species. So, shedding rates, again, it's based on, could be spawning activities, it'll be higher reproductive status, etc. And then it degrades at different rates, like I said. So on the right, you have a degradation rate going up, and then at the bottom is temperature. Some studies show it's a 1-to-1 relationship. So as temperature goes up, it degrades faster. But some people show that it's the opposite, or it's at low temperatures, there's a low degradation rate, and at high temperatures, it's low. So you have to take into account a lot of different things when you're trying to interpret what these results mean. Is it live or dead? A dead fish will release DNA, a live fish will. So when you get a detection, is it alive or is it dead? When was it there? So a little bit about the sensitivity of this. In a 9-liter aquarium, there was a study done with round goby, and it was done at 2 temperatures with different abundances. So the top 2 graphs are at 19 degrees, so at the same water temperature, and just one goby was in there, and then 3 gobies. And you can see that the same temperature, the one that had more goby had more eDNA concentration in it. So that tells you that potentially it could be correlated with abundance of a fish. So you might be able to go out and say, this is a highly abundant population. And then at temperature also affects it. So at the bottom 2, you have a higher concentration at a higher temperature than at the lower temperature. And the other thing to note here is that over time, the shedding rate or the eDNA will go up right away, and then it kind of balances off, and then it goes down. So there's a lot of different interactions. Again, it could tell us about biomass or abundance. There are mixed results here. Again, some people show a 1-to-1 relationship. Some people show this nonlinear relationship in this graph up here, where it goes up really fast And then it levels off. So everyone's probably wondering, false positives. That's a big thing that people are always wondering. What if you get a detection and there's no fish there? And it was a problem, more of a problem back in the day when it first started. There was a lot of contamination issues. So in the field, maybe we're in the cause in Chicago and we're sampling one area and we get DNA, it's all on our boats, all over our boat. And then we go over to Indiana and sample over there. Well, the DNA is coming off the boat and it's probably getting in our samples. So it might show that we have carp in Indiana when we don't. And so that was a problem at the beginning. We have come through that, which I'll talk about in a second. But then there's also potential contamination in the lab when you're working up, doing the genetic stuff with the different samples and whatnot. But there's a lot of DNA that's out in the air and on the hands and stuff. And so we have come a long way with our process, our methods, our procedures of not contaminating things. So false positives nowadays, at least for Fish and Wildlife Service and the methods that we use, are very, very minimal. One other thing is we get the DNA sequence that identifies it as a specific species comes from a general bank of DNA information. And so anyone can post on this site. So it could be, you could do it. And we wouldn't know, if we could replicate that or not. Or a geneticist from the Whitney Genetics Lab could put something in there. And so we have to verify this information that's in this bank of great information. So this is just a dashboard of what the monitoring of eDNA for all of the Great Lakes. And these yellow dots are basically where we sample throughout the Great Lakes for DNA. And it's just a dashboard. It kind of tells you how many samples we have over time. So we have over 40,000 samples since 2013. And aside from Chicago, which I'll talk about, there haven't been any detections of silver or big head carp. So this is the Bible here that we use. It is our guiding document. It's called the Quality Assurance Project Plan, or the QAP. It talks about all the methods that go into taking the water samples, decontamination, working them up in the lab, so we can defend it, what we do, and make it consistent and usable. So how do we choose where we sample? How do we choose how we sample? How many samples do we take? So based on silver carp and big head carp, we choose areas that would be good habitats for them to reproduce in. They have very specific requirements to reproduce, and it's in rivers, large rivers, with a certain amount of length for the eggs to transport and develop into larvae. That could be 25 kilometers. It could be 100 kilometers. So in Lake Michigan, we don't have a lot of rivers that are very long to support that. So we chose a few rivers in Michigan, the Kalamazoo, the Grand, and the St. Joseph River. We have Milwaukee River, and we do the Fox River. And those are also what we call hotspot locations, where there's a high probability based on different vectors of introduction for invasive species. So we look at the life history of the fish. We also look at when should we sample? Well, they tend to spawn in the springtime, and that's when they're going to congregate. That'll give us a higher detection of them. So we sample in the spring, but we also think about water temperature. We want a moderate water temperature, and that all just kind of aligns for silver and big head carp in the springtime. And we only sample in riverine environments, but lake environments are also sampled as well. And one sample per 0.01 kilometers. So you're basically sampling very, on top of each other almost. So this is where we sample. And again, they're potential spawning habitats. And they'll spawn during the increased, hydrograph at moderate temperatures. And so we've sampled these 5 areas since 2013 and haven't gotten any hits yet, except for this past year, which I'll talk about. So the gear disinfection is a huge, huge thing. We do it before we collect, we do it after we collect, we do it during collection. You've got to break down that DNA on the boat, on your person, on your life jacket, on your boots, in the truck, where you sit in the truck. I mean, it's crazy, but we're doing it because we can say almost to certainty that there is no contamination. We have dedicated gear that's only for our eDNA sampling. And it is crazy. We use gloves. We probably go through thousands of gloves in a week. It's crazy. It's dumb, but we have to do it. So we take water samples. I've been talking about this. So we have these 250 milliliters of water that we take from the surface of the river. And we have 5 vials down here. And we'll, scoop it up. And so there's 5 replicates per sample. And then, our pattern of sampling, where we sample will depend on the habitat, of course, but this is an example of a grid that we sampled below a dam. And then we also have what we call field blanks. And that's just a quality control, contamination step. It's pure water. And if that shows up to be positive, then there is a contamination issue. We have these mobile labs that are pretty sweet. We're able to process preserve our samples in the boat, at the boat ramp or in the parking lot of our hotel, because they have to be preserved in 24 hours. And driving this thing in Chicago at rush hour is not fun. So we preserve them in these laboratories. We centrifuge them down, and then it comes up to like a little pellet at the bottom, and then we preserve it with ethanol, and then we send it off to the Whitney Genetics Lab in La Crosse, where they do their thing. So our program in Lake Michigan, these are all the places where we've sampled over the years since 13. Over 16,000 water samples have been taken. And as I mentioned, we didn't have any detections of any silver or big head carp until last year. Milwaukee Harbor 2021. We sampled there in May and we sampled again in June. We took 100 samples each time. And then results got released in August that we got a positive detection from our June samples of a big head carp. So that was there. And this is Lake Michigan on the right. Then you have the confluence, and then it goes up into the Milwaukee River and down to the Kinnickinnic River. So this positive detection was right there, close to the confluence in the Milwaukee River. So we had one detection. So one sample out of 100 turned up positive for a big head carp. So what did we do? We worked with Wisconsin DNR and decided what to do, what we should, how we should proceed. So we decided that we should take some more water samples and see what that turns up. So we took an additional 150 samples in September, and we got the results back in a couple of weeks, and we had one positive detection for this time invasive carp. So it could have been big head or silver. It wasn't specific. This time It was in the Kinnickinnic River, but not very far away, but still in a separate river. So now what? We have 2 detections. That's not very many at all. What do we do? What does this mean? So we went out and we tried to go get a live fish so we could say, hey, here's one, you know, they're there. But we didn't catch any. So we went out for a whole week, did a lot of effort, and didn't get any fish. So that was good. So what does that mean? It means that there was DNA of a carp in the water at some point in time when we sampled. So when is unknown because it degrades over time. So there is a time frame. You know, it could be 5 days or 10 days, but we still don't know how long ago that fish could have been there. We don't know if it was alive or dead. You can't pick that up. You got the DNA, so you don't know if it was alive or dead. We don't know if it was moving through or if it was, you know, kind of a resident there. The DNA could have been not a live fish. It could have been transported by the boat, being on a boat that was in carp infested water, say Chicago earlier in the day. A bird could have dropped poop and had carp in it. I mean, all these sound crazy, but it happens. Wind currents. And again, Chicago is very nearby there, and there's a lot of carp in Chicago. In a bird, it's been shown that carp DNA can last eight days in the gut of a bird. So if you got lots of birds, lots of pelicans pooping in an area, it could come up as positive. Now, I'm not saying that this is what happened in Milwaukee, but we have Bubbly Creek in Chicago. So this is another thing that happened that could explain why we got a positive detection. So in Chicago, it's a creek off of the Chicago River. It's not really a creek, it's more of a canal, but there were a lot of positive hits. So every single one of these blue and purple and red is a positive hit, positive detection. But it was collected downstream of a pumping station during a flooding event, or during a pumping event. So they followed it up and they sampled the sewer lines that go into the pumping station. And all of those were positive. So then you start thinking, well, what's going on? And it turns out that Bubbly Creek is right below the Chinatown district that sells carp in their fish markets that go into the sewers, that go into Bubbly Creek. And they're readily eaten there. So again, in the sewers, in the creek. That is why we got so many detections in that area. So we take that back to Milwaukee. Not the case. There's no runoff. There's no, sewage plants anywhere near this area. The other thing to look at here, and in my opinion, it was a live fish, but that's just my opinion. The reason I think that is because it was in 2 different rivers. So it had 2 different inputs. Now they were close, and these are migratory fish, but the fact that we got 2 different detections is very improbable if it was contamination. So that's the quick and dirty of it. The main thing is eDNA should not be used on its own. It should complement our traditional sampling. Always fish in hand is better, and to have fish in hand, because there it is, it's right here. And we continue to refine our sampling every year. We sample different areas, we sample different different numbers of samples in an area, and we try to refine it and get better at detecting these fish if they're there. So that's all I have. I hope I didn't bore you too much. I close it.
Question. I got one, Mark. We had a positive test here probably about 10 years ago.
Yeah.
It was taken by Potawatomi Park. At that time, I think it was eventually determined that it was from a different location because it happened about the time the bass boats came here
Mhm.
and the pelicans came.
Mhm.
But they said they were going to increase the number of tests and the frequency of tests. Is that still going on?
Oh,
Yeah. We, in response to that, we did go out again and sample the area again, with some effort, some good effort, and we didn't get any more. But looking back on what you just said and potential contamination issues and stuff, we kind of have thought that they're probably not there. And so we wait.
And so in the meeting that was conducted, I asked a question that, because I have swam in Lake Michigan, could you find my DNA?
Mhm.
And the answer was, if we were looking for it, we would find it. There's 6 quadrillion gallons of water in Lake Michigan.
If we were on top of you, yes.
You would find my
Yes.
DNA.
I would say if you were swimming here and I was taking water above you, I would probably be able to detect you. Yeah, it's kind of scary.
Question.
Yeah.
How do you take into account that the water is constantly moving? You know, the rivers, you had a couple of samples at the Milwaukee River. I mean, that water flows constantly.
Mhm.
You know, so you're looking for DNA. I mean, it might be a fish 10 miles away.
Right.
And that DNA has floated down here, now you pick it up.
Yeah.
You-
Yeah, it's hard. You know, you can't say exactly where that fish was, just that it was in this water body. And, you know, there's lots of really smart statisticians out there that do some modeling and can say, oh, based on this water flow, this is how far the eDNA can go down. So there are ways to somewhat predict maybe where that fish was or where the DNA was in the system, but no. So in terms of Milwaukee, you're right. It could have, the backflow from Lake Michigan could have gotten to where those samples are. So they could have been from Lake Michigan, yes, when they're flowing backwards or whatever. And, you know, they're migratory fish, so they're going in between, fish are going between the Kinnickinnick and the Milwaukee. It's just that those 2 hits were not below an effluent or a sewage plant. That's all. So yeah, we can't say, we know that there was DNA somewhere in that vicinity.
Yeah, it tells you there's a fish
Yeah,
somewhere.
exactly. There's presence somewhere.
Another question.
Mhm.
Is there a way to tell the degradation of the DNA? In other words, you know, it starts out fresh and then after a while, you know, you're just barely picking it up. You
Right.
know, what you're looking for. Would that be another way of saying, hey, this thing's been in the water for a
Right.
long time?
Yeah.
And it probably came from somewhere else.
Yeah. And there's lots of studies that have been done on that, and specifically for carp. And so based on this size carp in this water, like a tank, mostly a tank experiment, it will go, you'll be able to detect it for 8 days. And then after 8 days, you can't detect it anymore.
But
So,
again,
but
is
in the
there a
wild,
grading? Is there a grading for the
Mhm.
DNA? Like,
Yes.
this is DNA at 10%
yes,
effective versus 90?
yes,
Okay.
yeah. Very smart people do that.
they do that on the genetic side of it,
Yes,
right?
yeah.
So at the Whitney lab, you know, they can get some idea of how much DNA is there and how old it is.
Yeah, so DNA is measured, it's copies per liter. So in a liter of water, a number of the genetic sequences are in there. And so that varies based on degradation or the size of the fish or all of that other stuff.
So if there are any questions on the Zoom, from the Zoom folks, you can unmute yourself and ask them and we'll be able to hear it here.
I got one more. Because Green Bay and of course Sturgeon Bay has been projected as being ideal habitat for both those types of carp.
Yeah.
Is that a place that you intensify your testing or not?
We do traditional sampling, a lot of traditional sampling. So we electrofish, we net in Green Bay. We haven't done Sturgeon Bay in a while, but we do Green Bay every year to complement our eDNA that we do. Milwaukee, for example, next year we'll probably do more sampling there or more additional samples to see if we can detect again. But you know, we work for the states, so we do what they request us to do. And right now, Milwaukee is where they want us to focus some of our efforts. And so Green Bay, for example, there was a fisherman last year or 2 years ago that, not a fisherman, he was a kayaker, and he claimed he saw a big head carp in Green Bay, in Dead Horse Bay. And so we went out and we sampled the dog snot out of it, and we didn't find any. And you know, based on that, we can't say no, it wasn't carp, but it more than likely was a buffalo, because we got a few of those, but we can't be certain. But we do go out and focus efforts if we have a reason, I guess. Yeah.
If you do find any positives in, let's say, the Chicago area, that's where they're closest, is there a plan as to how to attempt to control?
Yes, there is a very large scale plan of action. There's a lot of people that we have to contact first and let know. So the states need to know, the state DNR folks need to know, various working group committee, Asian carp committees need to know, and then there's going to be a large response. And this has already happened in the past. They've gotten a few detections, and I can't remember if they called it the carp rodeo or something. Silver carp bullet was what it was called a few years back. And there was like 10 boats on the water, traditionally sampling, electrofishing and whatnot. And then they took more eDNA samples. So it is a big deal when we do detect these fish. Yeah. And we didn't get any, by the way, with silver carp bullet.
So there was a question on chat. Has anything breached the barriers yet?
Yes. Yes, so we have found some DNA and a couple live carp on the other side of the barrier, close on the Lake Michigan side of the barrier. But we again have done follow-up sampling every year and haven't found any since 2017.
And they were not found in Lake Michigan.
No.
When you do sampling in the Milwaukee River, how many days or whatever would you avoid a rain event?
Oh, it's a good question. It's a great question. What is it? If it rises a foot in a day, we will not go out. So that's a pretty significant event. But yes, that does definitely affect the DNA in the water, dilutes it and whatnot. So we will not go out when there's a good rain event.
And even when you sample the rivers, sometimes you're looking at eddies or
Yes.
places where scum's collecting, you know, that's where that floating eDNA will also collect there. So they kind of look at those places instead of out in the middle of the channel and that kind of stuff.
Yeah, we're trying to sample backwaters and along shore. We are doing studies on the amount of DNA that's found in the mid channel, in the side channel, in the backwaters, and yeah, the dilution in the mid channel definitely drops off that DNA.
So Gary, back to your question about do we sample in Sturgeon Bay and Green Bay, or do they sample? It's not.
It's not you anymore.
We anymore. But basically where all these yellow dots are is where they sample every year.
Yeah.
So,
But
so
Sturgeon Bay, no, we don't do that.
we don't do that
No.
anymore. Sorry.
But that was the event you're talking about when we sampled there. I think it was in,
So is there a plan to do it again for
not, no.
So there's also another question, does the lake itself offer any protection from the spread should they get into the lake?
Depends on who you ask. So there is thought out there that the lake itself will not provide enough food for them to survive. I don't necessarily agree with that, but the lake itself is definitely less productive than the rivers where they are. And so they'll more than likely, they'll be moving from say Chicago up to Milwaukee to get to Milwaukee to get to the river. So they'll be moving in the lake proper, but probably not residing in the lake. So that would be a barrier potentially that they have talked about in the past, that there isn't enough food for them in the lakes. But no other barriers, like physical barriers.
And so, this all got started when I was still working, and basically the Fish and Wildlife Service, because they could deploy the same program across each of the Great Lakes, have been doing this eDNA sampling and so on. When you do get a hit, you can imagine the jurisdictional issues about if there's a carp here or not. And so when you do get a hit, basically then we turn to the management authority in that area, and then the state will sort of dictate what kind of response there is. We will support, the Fish and Wildlife Service will support it, especially follow up with more sampling with eDNA or even traditional gear and so on. But the actual extent of the search and everything is really primarily up to the state agency where it occurred. And there's been lots of discussions across or with each of the agencies exactly how they want to handle that. So it's a very sensitive issue. You can imagine if there's really a carp found, because I know within the Fish and Wildlife Service, they've gone to the extent that they've stockpiled rotenone. So that if there is a positive fish found in other fish in an area, that the Fish and Wildlife Service can then offer this rotenone in a very short order to try to nail it down as soon as they can. So the advantage of eDNA is that if there's you can detect very small amounts of DNA that may tell you a fish is there. Sometimes you don't know if it came from a bird or a boat or everything, so that it's got its drawbacks, but it's the quickest way to know, okay, we got to look further. And if there really was a fish in the Milwaukee River that stayed in the Milwaukee River and you followed up sampling, you're going to see it again and again. And that necessarily hasn't happened in the Milwaukee. So it's up to the state then how much effort you really want to go look for what could have been DNA from a bird or something like that. So, the good news is that there's somebody watching with a very sensitive tool. Sometimes it's a blunt tool, but it's a lot more information than we had without it. And the Fish and Wildlife Service is able to develop that kind of program, for the Great Lakes.
And another thing that we're doing, that we're starting this year or next year, grass carp are a very big problem in Lake Erie. They are reproducing, they are spreading, they are becoming a problem. We are starting to find them in Lake Michigan here and there, and they're reproductive. And so we haven't found any, larvae or young yet, but we just know that they're able to reproduce. And so we're finding them. We found one in Milwaukee. We have one in St. Joe River, up in Muskegon Lake, down in Indiana Harbor in Indiana. So they're showing up, and we don't want this to happen like what happened in Lake Erie. So we don't know where they are all the time, like all over the lake. We don't know where they are, but we can use this eDNA as a tool to focus and try to find where the hub is. and maybe combat them before they become a problem.
Any more questions?
Thank you.
If not, thank you, Cari-Ann. And thank you all for coming. Don't forget, March 17th, you'll get to learn about suckers in the Great Lakes, and it'll be a very entertaining presentation, I guarantee it. So anyway, please make it, and thank you very much for coming. It's much appreciated. And you can sign off on Zoom then too. Thank you all.